Hello friends, welcome back to my channel, and today in this video we are going to study about gene mapping using molecular markers. So with molecular markers we have to map the genes. So RFLP is one of the markers. As the name suggests, restriction fragment length polymorphism. Polymorphism means more than one form. What form we are talking about? Forms of length of fragments we are talking about. And how we generate these fragments? By using restriction enzymes. So we are composed of chromosomes. When we apply any restriction enzymes, so as the name suggests, restriction enzymes means these are the enzymes which are also known as scissors because they cut our DNA molecule at a specific location. So when we use these restriction enzymes, they'll cut our DNA molecule at a specific location wherever the site for this enzyme is present. Say for example, two persons are there. They both have their own DNA. Now, when we use the restriction enzyme A, say for example, it can cut in person A at this position and in person B at this position. So if it is cutting at this position in person A, we'll get the fragments like this. Whereas in B, we'll get the fragments like this, two equal fragments. Correct? So that means these are the different forms of fragments which are generated due to the application of restriction enzymes. So because we inherit the gene from our parents. So, for example, if A is married to any person C, so their child will also possess the site of restriction enzyme A at this position only because it is inherited from this parent. So because we inherit the genes from our parents, so in our parental DNA, wherever the site for these restriction enzymes are present, these sites will also get inherited in our body because the whole genes are getting inherited. So this is called as restriction fragment length polymorphism because we are getting more than one forms of the length. Length of what? These fragments that are generated due to restriction enzymes. And because these DNA molecules are inherited from one generation to another generation, therefore, the site for these restriction enzymes also inherit from one generation to another generation. So therefore, if parent A is getting cut with restriction enzyme 1 at this position, the offspring of A will also have the site of restriction enzyme at the same position. So with the help of this, we can easily map the gene found in our body. So restriction fragment length polymorphism is used not only for gene mapping, but also it has multiple applications like parental disputes, in crime investigation, forensic, etc. So we are not going in deep about this parental and crime. This we will study when we'll study about the molecular markers in biotechnology subject. Now, in genetics, we only have to use the RFLP to map any gene presence. So these differences are inherited and hence can be used for mapping. Now, we have to map a gene for a disease called Huntington disease, which is an autosomal dominant disorder with the help of a RFLP marker. Now, how we can do that? Now, here two pedigrees are available in front of us. If we see the pedigree present in the left hand side, the pedigree 1 and this is pedigree 2. If we see the pedigree 1, in generation 1, this female parent is having the gene B and it is also having the recessive allele of this disease, smallest. Whereas the male parent or the father has the gene capital H and small h, that is one dominant allele for the disease, and also carry the gene A and C. So the mother have only B gene, whereas father carry A and C gene. Now, because mother is having only B gene and the small h allele, so we can say that this B gene is linked with small h allele of the disease. Now, if we see about A and C, so A can be linked with capital H also, small h also. Similarly, C can also be linked with capital H also, small h also. Now, if we see the offsprings, here we can see A and B is inherited and the offspring is disease free. Here what we can see, small h, small h. So if we are considering that B is linked with small h, so one small h is because of this B gene, because B is inherited, therefore small h is also inherited. Here, according to this, the A gene should be linked with small h, but if we see here, for B small h, but here along with A, capital H is inherited. So in one progeny with capital A, small h is inherited, whereas in another progeny with A, capital H is inherited. Similarly, if we see the B in the second offspring, CB, what is inherited? With C, capital H. Here, the fourth offspring, what is inherited with C? Small h. So that means here the RFLP gene position is not linked with the particular disease. That means these two genes are independently assorting. We can see independent assortment here. So these genes are not linked together. Whereas in the pedigree 2, again the mother consists of gene B and small h allele, whereas father has both the genes A and C and both the alleles. Now what we can see here, capital C in the offspring 1, capital H, capital C, capital H, again C, capital H, again C, capital H. Whereas with A small h, A small h, A small h, A small h. That means here we can clearly say that gene A is linked with small h allele, whereas gene C is linked with capital H allele and gene B is also linked with small h allele. So here the RFLP pattern of the gene is linked with the particular disease. So because this gene is linked with this disease and this is the pattern of RFLP, that means if we use any restriction enzyme, it will cut at this gene position. So because we know the position of this gene, because we have restriction enzyme available for this gene, now with this gene, because H allele is also linked, so we can easily take in account the position of small h allele. So by this way, we can easily tell that where is the position of the diseased alleles. So RFLP markers are then used to as an indication for the presence of disease causing locus. Now this is one question. This parent, female is disease free and male is diseased. Now they are having 12 offspring. Now for two restriction enzyme, the patterns are given. Now your question is, first of all, you have to determine that whether this particular disorder is linked with any of these restriction enzymes or not. If linkage is there, then you have to guess the recombinant progenies. And if recombination is observed, then you have to calculate the map distance. Clear? Now how we can solve this? First of all, this is parent 1, this is parent 2. Parent 1 is normal and marker for normal is smaller. This pattern we can represent it as smaller. P2 is diseased and RFLP marker is also present. Now offspring 1 normal, this is also no marker. Offspring 2 diseased and the marker is also present. Offspring 3 diseased but no marker. Normal, no marker. Diseased, marker, diseased, marker, diseased, marker, diseased, marker, normal, no marker, normal, marker, diseased, marker, normal, no marker. What is our parental type? NR and DR. Capital N, small r, capital D, capital R. Now, if we see this offspring, what we can find? Offspring 1, parental type. Offspring 2, parental type. Offspring 4, parental type. Let me circle it for you. It will be easy for you to understand. This is parental type. This is parental type. This one parental type. These are all parental types. This is parental type, and parental type. So what we can get? even in case of linkage, we know the number of non-recombinants are always more compared to the number of recombinants. So here we can see that parental types are more, that means non-recombinants are more and recombinants are less. Two are recombinants, which are without circle. So that means with restriction enzyme 1, this particular disease or disorder is linked. Likewise, you have to find for restriction enzyme 2 also. Correct? So step 1 is to see whether the disease allele is linked with marker or not. Here, diseased allele is shown by A asterisk, whereas I showed you with capital N. So whatever way you want, you can do. Step 2 is how to find out it is linked or not. For parental 1, I showed you normal and no marker. For parent 2, diseased and marker. So on the basis of this, you can find for restriction enzyme 1. I already showed you first one is normal no marker, second one is diseased and marker. So like that, the way I showed you, you have to find out for restriction enzyme 2 as well. Now, how to decide whether the genes are linked or not? As I told you, more number of non-recombinants than recombinants means linked gene. Recombination can never be equal or more than 50%, that we know already now because we learned it from quite few videos. Clear? So for restriction enzyme 2 also, the way I showed you, you have to perform the same thing to see whether the second restriction enzyme is also linked with this particular gene or not. Correct? This is the parental type 1. Now, after that, what we have to do is with restriction enzyme 2, we found 2 recombinants, rest all are non-recombinants. So as I said, the genes are linked. For restriction enzyme 2, you have to find it out. Now, we have to count the number of recombinants. So out of 12, how many recombinants we got? 2. Fifth step, you have to calculate the recombination frequency because you have to find it out the map distance. So map distance is MU. MU means 1% recombination frequency means 1 MU. This also we know now. So you have to find out the recombination frequency with the formula. And on the basis of that, you have to tell the map distance. Clear? So you have to find out for restriction enzyme 2 everything, and for restriction enzyme 1, I already told you number of recombinants 2, total number of progeny 12. So the formula is pretty clear. So for 1 I told you, for 2 you have to calculate, and do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.